So this is a structure of DNA. This is a phosphate. This is a deoxyribose sugar. And then this is a nitrogen base. This is specifically adenine. They come together to form a nucleotide. So that's adenine, of course, guanine, cytosine, and thymine. And both adenine and guanine are purine. And thymine and cytosine are pyrimidine. And they come together to form complementary base pairs. The phosphate and the deoxyribose sugar are hard to take apart, which is to symbolize a covalent bond to where they aren't easily. Why am I struggling so bad? Aren't easily taken apart. But the hydrogen bonds between the cytosine and the guanine are very easy to unzip. And because I didn't say it before, this is the backbone to where they hold the DNA together. Here we have a codon where it's just 3 nucleotides stuck together, and then when you stack those are genetic codes. Adenine and thymine have 2 bonds, while guanine and cytosine have 3. The DNA helix is shaped in such a way so that it can be squished down and stored in the nucleus as a supercoil.
During replication, the genetic code, or the DNA, is going to be unzipped by the helicase enzyme. Then, it's going to break the hydrogen bonds between the complementary base pairs, and then you have a DNA polymerase, which is the enzyme that is going to connect together those complementary base pairs. As the DNA is unzipped, the leading strand is going to build continuously so that the sides is going to be done differently than the other sides. On this side, this is called the lagging strand. With the lagging strand, you have a primase. This is going to come in and lay down a primer. So the primase lays down a primer. Then you have the preliminaries that will come in and build in the opposite direction. And then you have another primer that gets laid down, and then the polymerase comes back in and builds the next section. Those sections are called ogonosacmi fragments. So you have 2 ogonosacmi fragments that are connected by a primer. Now that you have those 2 built, those ogonosacmi fragments need to be connected. You have a ligase enzyme that is going to then get rid of that and place down a nucleotide that should go there, which connects the entire sequence, so that in the end you have 2 identical strands of DNA. This process is the start of mitosis and neomeiosis, and in order to make sure they're identical. In transcription, the DNA goes into the polymerase, in which the polymerase is going to build the mRNA sequence. It does that by matching up complementary base pairs. As the polymerase goes through, the mRNA is built, continuing to match up those complementary base pairs until finally the entire mRNA sequence is made. Now the mRNA sequence is matched up on this side with the non-coding strand. It's a template that matches up the pieces. Now on this side, this is called the coding strand, because if you look at the mRNA, it is exactly identical to the coding strand. So by matching up the non-coding, you have created this actual coded sequence for what you want to build. Now, the mRNA is made up of 2 types of sequence. Introns are pieces of info that are useless pieces of info, and those are going to be taken out of the mRNA. The useful pieces are called exons.